A transgenic organism is one in which a foreign DNA molecule, a transgene, has been randomly incorporated into its genome, whereas knockout and knock-in mice have targeted DNA insertion. The DNA inserted into the organism may be from the same or different species. In creating a transgenic mouse, recombinant DNA technology is first used to generate transgenes such as constructs consisting of a promoter, a cDNA, an intron, and a polyadenylation signal in plasmid vectors, or genomic DNA in bacterial artificial chromosomes. Genomic clones in back vectors may be inserted as intact circular molecules, while constructs in plasmid vectors are excised and inserted as linear DNA. The DNA is purified before transgenesis. The transgene is introduced using the microinjection technique into the male pronucleus of fertilized eggs obtained from crosses between wild-type mice. The random integration of genes into the genome may occur at a single locus or multiple sites. This random integration may disrupt endogenous gene expression. In preparation for embryo implant, the female mouse is induced to pseudopregnancy. Once embryos have developed from the microinjected eggs, they are implanted into the pseudopregnant female mouse. The pups subsequently born are tested to see which, if any, have the transgene incorporated into their genome by taking a small tissue sample from the tail and analyzing it with PCR and gel electrophoresis. Mice with a transgene are bred with wild-type mice to check that the transgene can be stably transmitted to the next generation, then bred to establish a line of mice that carries the new gene. Transgenic mice lines may be created to study diseases, as well as used in testing drug efficacy and safety.